All right, so we've talked a lot about how to think about speciation of acids and bases in solution as a function of pH. So we've thought about it from a couple different perspectives, from first principles, systematic treatment, through approximations. We've thought about both monoprotic and polyprotic systems. We thought about, well, if you've got both a weak acid and its conjugate base, or a weak base and its conjugate acid in a system, we call that a buffered system. So we've sort of covered all the bases of what species dominates as a function of pH. And really, then the natural sort of end of that discussion is thinking about, well, what if we do that on purpose? What if we change the pH on purpose? And that's what a titration is. And so we have all the tools now to compute the different parts of a titration curve. But before we jump into some of the tedium, which is dealing with titration curves, I want to give you some framing, because I've mentioned that I don't do titration curves, but that's just the nature of my science. It doesn't really need that, and it never has. And so the last time I did a titration was as an undergrad working on environmental chemistry research, where you still often need to do titrations because you're titrating complex natural waters, and it's the only methodology to measure what's called alkalinity of a system. So that's really common in environmental chemistry to continue to do that. But it's also still really common in biological chemistry, chemical biology or biochemistry, whatever you want to call it. It's all the same. This example that I grabbed is real data published from a group where they were titrating a specific enzyme. And an enzyme, for those that aren't biologically disinclined, haven't taken a bio course or are not interested in that, enzymes are these natural proteins being these polypeptides, where you've got a bunch of amino acids all bound together through particular bonds. These things all fold up, and they're fairly large molecular weight objects in solution. And what's interesting about them, if they're categorized as an enzyme, what they do is they help accelerate the rate of certain reactions in a biological system. And they do that by taking a reaction and usually spatially orienting it in a way that it can't orient in a natural way. And by doing that, it allows a separate pathway to completion, to products, thereby lowering the activation barrier for the second or the alternative reaction pathway. What's fascinating about enzymes is they do what we as chemists wish we could do, and they do it really naturally. They make it look easy, right? They can do these amazing carbon-carbon bond transformations in water at, biological pH 7. As chemists, we give Nobel Prizes for people who do that at elevated temperatures with platinum catalysts to do these crazy cross-coupling reactions. But these natural enzymes, they do it without all of that. So what a lot of biochemists, they want to understand why it is that a system like this behaves, and so there's sort of two approaches. One is to just sort of figure out what the structure of the peptide or the protein is, and we can do that typically with crystallography or free electron laser diffraction, amongst other things. There's other structural probes as well, but those are the typical things that we do. But it's really difficult to relate structure of one of these large biomolecules with its function. And so to do that, you don't want to crystallize it, because that locks everything into position and assumes everything's static in space. And so that's usually a bad assumption for biological mechanisms. And so what biochemists do often is they actually titrate the peptide or the enzyme itself. And the data I'm showing you is real. This was for the titration of a particular enzyme. I can't remember. This is ribonuclease. So this is the titration in ribonuclease. This is a couple decades old at this point, but the idea is the same. You can do this with any enzyme to assert what the function is. And so what they do is they toss some concentration of this into a cell, and then they adjust the pH to the isoionic point, which typically it naturally does in a buffered system. the isoionic or the isoelectric points, which are, they're slightly different, but not different enough for us to actually define them differently in this class. They're essentially the pH at which the overall charge on the molecule, in this case the entire enzyme, is neutral. The average is neutral. And that makes sense because what is a protein but just a bunch of amino acids that are bound together? Amino acids have both an amine and a carboxylic acid group, so you can deprotonate the carboxylic acid group, that makes it negative, or you can protonate the amine group, that makes it positive. If you have both of those things present, you have a zwitterion, which means you have a positive and a negative, but remember you've got a whole bunch of amino acids on this structure, all with a bunch of different pKa's. So the pH then is sort of, or the isoelectric point is sort of this average where the net of the entire molecule, the entire enzyme, is 0. So you start there, and you can see that on the x-axis you have acid being added per molecule, and then base being added per molecule from 0, 0 being where the isoionic or electric point is in the system. And I think this thing has upwards of, like, I don't remember, 20 or 30 different residues that are titratable, meaning you'll see when we titrate, a monoprotic or a diprotic acid, we have these sort of 2 clear inflection points. But when you've got something like 30, the inflection points all sort of blend together, and that's why you get this sort of non-distinct titration curve. But by doing a little bit of math, taking the derivative of this, you can actually discover where the inflection points are for the system. And what that tells you is it gives you a direct measure of what the pKa values of these residues are on this protein as it's functioning in real space. And the importance of that is that it gives us some insight into the actual behavior of the enzyme. How does it do that? Well, imagine the sort of example that's shown here is you've got tyrosine, which is a common amino acid, and you got 2 of them. One is located on the periphery, on the external portion or fold of the protein, and that's said to be easily accessible by base, right? Base can come in, and the hydroxide doesn't really need to diffuse inside of the protein itself, so it can grab that protein easily. And this tyrosine, probably in a titration, would have a pKa very similar to what a theoretical pKa value for tyrosine would have. But at the same base concentration, where you remove that proton from the external tyrosine, the internal tyrosine inside of the fold remains protonated. And what that tells us is that the hydroxide has a much harder time accessing that. And so the apparent or the experimental pKa for this tyrosine residue is very different, can be orders of magnitude different than the tyrosine on the external part. This gives us a lot of information, because if we just sort of operated based on structure, we would say, okay, tyrosine there, both have the same pKa, but that's factually incorrect. And so this is really fascinating, right? Because pKa's, we think of these things as these constants, right? But those are assuming that there's nothing that's impeding access to that molecule. And so you've got probably physical sort of steric effects that are preventing hydroxide from diffusing, but you also probably have other electrostatic effects that are impeding the diffusion. There could be other negative moieties inside of that fold, and then you have a negative hydroxyl group trying to diffuse, so you have electrostatic repulsion. There could be lots of things at play that that start to be exposed through a titration like this. So I go through and I tell you all of this just to give you a little bit of framing in the power and utility of titrations, even in the year 2020 when this video is recorded. So it may feel tedious to you at this point, and it is kind of, but it's for greater good, because experiments like this are still invaluable, and there's really no alternative way. There are folks in our department who do titrations, biochemical titrations often, and not only measure pH changes, but also then measure heat changes. So doing titrations in calorimeters. And that can tell you a lot about how different things are adsorbed in a molecule when you add certain amounts of, say, metal ions to DNA or RNA, and then you measure the heat change, whether that's heat going into the system or out of the system. You can associate different concentrations with different binding effects inside of these things. So titrations are really important.